So in this next lecture for the Bio 410, Bio 510 Bioinformatics class for California State University, Monterey Bay, I'm going to be talking about sequence alignment. So we'll transition a little bit out of statistics and start to look at how we might engage algorithm. And so today we're going to take mostly a dig into one specific algorithm, just as an example to show how algorithms can be used in sequence analysis. And we're starting on our journey of just overall sequence analysis, which there's a lot to cover. So we're going to focus in on BLAST today, on the BLAST program and approach and algorithm, and then spend a little bit of time thinking about the differences between local and global alignments. So let's talk about algorithms first, right? So there's kind of two ways we might, there's many ways, but here are two ways to define an algorithm. We have a procedure for solving a computational problem with a finite number of steps, or we could be a little more general and say it's just a procedure for solving any kind of problem, right? And so if we think about the first one, we can target in on a specific type of outcome, like finding the longest common subsequence between two strings. But if we think about the second case, we can see that might be more of a more general way of approaching this idea, and that we're looking more at kind of like the step-by-step flow chart or pseudocode or some more sort of general way of describing how to solve a particular problem that's not necessarily tied to a specific computational issue. And so I think it's just important to note that we use algorithms all the time in our daily lives to make decisions. They're built into our behaviors. They're built into the tools that we use. And so it's kind of a defining characteristic of how we navigate our realities. So just something to realize that algorithms are everywhere. They're around us all the time. And they're really important to how we approach our lives and our science. So let's take an example of what might a simple algorithm look like. So say we wanted to find the largest number in a set of numbers, right? We would just set up a series of rules, right? And the first one could be if there's no numbers in the set, then there is no highest number, right? We're going to add in, so we're going to start with nothing. Then we're going to assume that the first number in the set is the largest number in the set. And then for every other remaining number in that set, if it's larger than our current number, then we're going to consider that one to be the largest number. And we're just going to keep on going until there are no numbers left in our set. We're going to keep on iterating or repeating this process. And at the end of it, we would have found what is the current largest number of the set. Doesn't seem like all that interesting, but just realize that what I'm talking about here when I talk about an algorithm is a set of rules, right? A set of rules that I'm going to follow all the way to the end. And that's going to help me come to some sort of conclusion or outcome, in this case, finding the largest number in a set of numbers. One of the first examples and classic examples of the applications of algorithm was through the work of Alan Turing and the cryptanalysis of the Enigma communications tool that the Germans used during World War II. And if any of you have seen the movie about this, it's The Imitation Game, I think is what it's called. It's an excellent movie. It talks about this, but this is basically the first time we took it's kind of like a computer called a bomb, and Turing developed a process that was referred to as Ben Burismus, which was a process for which they used conditional probabilities, so sequential conditional probabilities, to infer what are the most likely settings of the Enigma machine. And so think about this. This is a really Bayesian approach, right? They're using conditional probabilities to infer what is the most likely settings for the Enigma machine, right? And so they had to figure out, they had this machine that had all these weird dials on it, and they had to figure out how to translate that into actual words. And they used language analysis basically to go through and do this process. Also tells you something about how useful language analysis is. A lot of the tools we use in sequence analysis are actually related to language analysis. Really interesting first approach. I definitely suggest reading up on it. It's a really interesting story, really important event for winning the war. We definitely suggest you guys to look into it more if you're interested. So we're not really interested in decoding Nazi missives about U-boat schedules and whatnot. We're a little more focused on some other questions, and these are mostly related to things like sequence alignment. This is a class in bioinformatics. We're going to be dealing with genomes and genome assemblies. Sequence alignment is kind of our first foray into sequence analysis. And we think about, what are we dealing with? What's the data we're dealing with? Well, we're dealing with the building blocks of life, right? And they've been translated into being an alphabetic format, into a string format of letters, right? So basically, we've got A's, C's, T's, and G's. We know that they are organized in strings, so consecutive placement of these together. And so, how can we leverage this sort of information structure to be able to actually look at biological entities? And one of the ways that we do this, one of the principal ways we do this, is by looking for patterns of sequence similarity and dissimilarity, and then using those patterns to tell us something about the history, identity, and even function of these entities. And so a classic way to do this is to take these sequences, and you have this example over here on the right, where you align them to each other, and then you can use the patterns of differences and similarities to build a tree-like structure that shows, that 4 and 5 sequence are the most similar to each other. They've only got this sort of one nucleotide difference here. And then we can compare that to 3, 4, and 5, and here we can see that there's an additional difference here and here, right? And then we can see that there are, and we can see these differences plotted off on this tree, which show why they sort of end up in this different clade-like structure, right? So we can see that certain changes, right, occur along certain branches. Other changes are related to other branches and their isolation of this lineage, right? This change is something that's shared among this group. This blue change is shared among this group. So we can see how these different changes can help to classify things, whether they're similar or dissimilar from each other. That's a classic thing we want to do in sequence alignment. So when we think about sequence alignments, there's a bunch of different ways we can define this. So one of the principal ways is thinking about the number of sequences. So in this case, we have both pairwise and multiple sequence alignment. So pairwise is just 2 sequence alignments. Multiple sequence alignment is more than 2 sequences. We have types of sequences, DNA, RNA, protein, all aligned using different mechanisms and algorithms. And then there's the scale, right? So we can either have a global alignment where we have to align all the letters and nulls in all the sequences, so from end to end, or we can have local alignment, which allows for an arbitrary length segment of each sequence to be aligned. So in other words, we're aligning just a segment, a portion, a locally important piece of the sequence, not the entire thing. And there's no penalty associated with the stuff that we're not aligning. So what constitutes an alignment? So over here we have some examples of some ways we might align some different words. It's kind of a good question here, like which one of these, are all of these alignments? Are some of them not alignments? If we think about the questions that are involved in alignments, we can say, yeah, of course, these look pretty similar. We've just got one letter that's different. Here we've got a gap that's different. Here we've got, some pieces that are rearranged. Then we start to get the very different words, right? And so we could say, well, is this really an alignment? Are we really align them to each other? Are these words similar enough that we would align them? Is this a better alignment? What about this disestablishment and descent case, right? How do I pick this alignment over this other alignment, right? So we can see that, in some cases it's clear what the alignment is. In other cases it may be less clear what the alignment is. And that at some point we may have to figure out, where there is some ambiguity, how we might pick one alignment over another. if we think about sequence alignment, generally, when we think about pairwise sequence alignment, especially, we always have two proteins or two DNA sequences. That's typically what we're starting from. And what we're looking to see when we do this is we're actually looking to see kind of how similar or dissimilar these things are. And when we do that, typically we're going to be calculating some value to measure that similarity or dissimilarity. And we refer to that as a distance, right? And there's got to be some objective part to it. And this is where algorithm comes into play, because we're going to have to decide on some rules that are going to allow us to do this in a rigorous fashion so we can figure out maybe how we might say one alignment is better than another alignment. The classic way that we're going to talk about how we do this, and that we do a lot, is we're going to talk about the dynamic programming BLAST algorithm. BLAST is something that is widely used for all of us. I'm sure all of you have blasted before. And what you'll notice is that when you do, say, a BLAST P search, you get your data back. You have this structure down here where you have, a query sequence and a subject which you've identified. You align them to each other, and you can see that they align by the regions that they share, right? And compensating for the regions that are different. So a couple key components of things to point out here when we do this, right? We're identifying things that they share, and then there are some other symbols that can provide some additional information, like these dashes indicate that this sequence is lacking sequence here, while this sequence has a sequence here. So we refer to this as an insertion-deletion region. And then because they're proteins, we might have certain proteins that are very similar to each other in terms of their chemical properties, and so we might identify those by something like a plus. When we align these sequences to each other, it's important that we understand what are the sort of entities that we're measuring that are going to form the components around which we're going to build rules, right? And that's going to form the basis of the algorithm, the components of the equation, so to speak, right? So things that we're going to be investigating often are things like matches and mismatches. So where do we see that sequences are the same and where are they different? And there's sort of an implied relationship here that where we see things that are different, we would think that they would either have some kind of shared function or shared ancestry, or both. And where they're different, we might see divergence in those things. In terms of gaps, sometimes we might represent that with this null character, this dash, right? And basically these are examples where there either has been an insertion or a deletion in one of these sequences relative to the other one. So maybe an extra codon added or deleted in one case. What happens is we'll take these patterns of mismatches and gaps, and we'll use them to calculate what we would refer to as an alignment score, which is going to be based upon the patterns of comparison between these two. And the alignment score is typically built out of two components. One is the substitution score, which basically scores the two sequences and how they align based upon the matches and mismatches. And the other one is the gap score, which scores the sequence alignment based upon how many gaps and how many on the gaps and how large they are that are in the sequence. Gap is kind of a tricky way to score sequences in this case. So we refer to them often as gap penalties, and this is typically subtracted from a similarity score in an alignment program. And these gap penalties can either be linear or affine. And so a linear gap penalty means that basically the same value is subtracted for each gap added to an optimized sequence. So every time I add in a dash, I subtract the same amount. An affine gap penalty means that the penalty has sort of two components to it. There's one penalty for opening a gap, and there's another penalty for extending a gap. So, in that case, it might be more or costly or harder to open a gap than it is to extend one. So there's different ways of scoring that. You can see an example of this over here on the right here. So when we align sequences and we assess their similarity, basically what we're doing is we're defining this similarity as the score of their highest scoring alignment, right? That's what we think is the sequence similarity between these two. How do we find the optimal alignment of these two sequences and its score, right? So this may not actually, you might think, oh, it's, we're sort of aligning these two words to each other. Well, it's actually a lot of work, especially the longer these sequences get, the more difficult it is to do by hand. And so doing it basically by brute force enumeration, there's just too many possibilities. Think of all these sequence positions, 4 different outcomes per position, and we think of the number of possible outcomes, like figuring out how to align them the best to each other, it becomes really computationally challenging. And so to do that, there's a technique called dynamic programming that is going to be applied, especially in the case of the BLAST algorithm. So dynamic programming with respect to global alignment involves is basically a method by which we can try to solve a larger problem by first solving a series of smaller partial problems, right? And computers are great at this, right? We give them a small, simple problem and get them then just to repeat it. We write that loop. And in this case, what we're going to be eventually calculating, right, is the similarity between two sequences, x and y. So basically, we're going to be solving for this equation where we're going to take the highest value of any of these 3 outcomes, right? And explaining how these are calculated, how we can solve for this similarity score, I think it's important that we understand that what we're doing when we're doing this is we're using as you see there's an I and a J, we're using a graphical matrix-like approach to considering these sequence alignments. So when we think about how we actually do sequence alignments and how we do a lot of sequence analysis, we're actually doing something where we're looking at the path through a graph or a path graph through a matrix or graph-like structure. And so a graph is anything basically that has nodes and edges, and we're going to look to see how we can align two sequences, x and y, to each other by navigating across this path, right? And the thing is we're going to try to find the best path through this path graph through this entire graph in order to best align these two sequences. And to do that, we're going to use this equation here. So remember, we're looking at the maximum score of any of these 3 equations, right? That's what we're going to use to sort of determine what the correct path graph is through. And to do that, we're going to use an algorithm for BLAST. The classic algorithm is something referred to as the modified Needleman-Wunsch algorithm. And one of the first things that you need to do is select a scoring rubric, right? So here we have the matches. Every time we have a match, we'll add 1. Every time we have a mismatch, we'll subtract 1. And every time we have a gap, we'll treat that as a -2, right? So we have some different scoring rules for our graph. And the first thing we always do is we fill out the first row and the first column of this graph with this matrix with what would be the gap penalties, right? So if I was to start at this position, I start at 0, and if I was to keep going across here and not have any matches, I would incur a -2, a -4, a -6, a -8, a -10, a -12, and a -14 penalty. And this is basically just me adding in gaps before I ever align these sequences. Same thing here. If I keep on adding in gaps, it's going to be -2 every time, right? So that's how I fill out this value. The next thing I want to do is I want to use this sequence. It's going to say, okay, what goes in this value is going to be the maximum of any of these 3. So if we think about how to calculate this similarity score, right? If I is for the number of row that you're in and j is for the number of column that you're in, if I move to this position, if this is the row 0, column 0 position, and I move to the row 1, column 1 position, right? I'm going to look and say, hey, is this thing a match, right? So if I go back to the I1, the similarity score for I minus 1 and J minus 1 position, so I'm going to at the 1, 1 position, I'm going to look at the 0, 0 position. So what's the similarity score at that position? It's a 0, right? And I'm going to add up the similarity score for the position that I'm at, the substitution score. So here that's going to be a match, right? Because G matches G. So this is actually 0 plus 2. So one of my scores is 2. The next thing I would do is look at the I minus 1 J position. So the row 0 J1 position. So that's minus 2 plus the gap penalty. So that would be minus 4. Or I could look at the similarity score of the I minus J1 position. So that would be row 1, column 0, right? It's minus 2 plus the gap penalty, which is minus 2. So this is minus 4. So the three possible answers I have when I'm at the 1, 1 position is either 2, minus 4, or minus 4, right? By looking back at these previous positions, right? And by doing that, I can see that 2 is the highest scoring value out of the 3 of them. And so I fill this position in this graph with the number 2. So as I fill out this graph, I can fill all of the other positions around it, right? So I can start here and say, okay, if I look up here, this is going to be minus 2. It's a mismatch, so that's going to be minus 1. So if I look diagonally, it's going to be minus 3. If I look above, I have 2, right? Minus the gap penalty, which is 0. If I look over here to the left, I've got minus 4 plus the gap penalty, which is minus 6. So either have minus 6, minus 3, or 2, or 0. And so 0 goes in this spot. So same thing here, right? If I look at this position and I try to fill it out, first I'll look diagonally and I'll say, okay, the previous similarity score was minus 2. I've got a mismatch, so that's minus 1, so that's minus 3. I'll look up, right? This is minus 4 plus the gap penalty, which is minus 6. Look over here, this is 2 plus the gap penalty, which is minus 2, so this is a 0, right? And then I'll do the same thing here. I'll look this direction, I'll look this direction, I'll look this direction, I'll keep the highest number. And I slowly go through and I fill out this entire path graph, right? And I end up with a series of scores like this. So once I have this series of scores, I'm going to do something that's called a traceback. And by doing a traceback, I'm going to try to find the highest scoring path through this sequence, and because it's a global alignment, it's going to be from the start to the end. And so by doing that, I'm going to be able to identify where are the matches, the alignments, and the gaps in the sequence. And so if I look at this sequence and how it would align, right, I can see that as I go from the C and the T here align, and then I go up, I've got a gap where there was an A in one sequence and there isn't one here. And then I'll have an alignment of T and G and T and A, right? And then I'll have another gap before I get over to the alignment of G, right? And so this ends up being my best alignment for this sequence by following this highest scoring process through this path. So local alignment is very similar to a global alignment, and a good example here is the Swift-Waterman's alignment. And basically what the main difference here is I don't have to align the entire sequences to each other, right? I just have to align the parts of the sequence that score the highest. And there's no sort of penalty for the ends that aren't going to align. And this was a big advancement in terms of speeding up this process, right? So now I no longer have to align the whole sequence, I can just do part of it. And basically what it did was it took the Needleman-Wunsch algorithm and it just allowed for some different processes here. So what I did was I basically just got rid of any negative numbers, right? So wherever there was a negative number, we would replace it with a 0. And what we then did is so we allow all the mismatches or everything to start at a 0, and then just record the highest scoring nodes, right? And then we allow that to trace back. And what it does is we end up with just much shorter alignments, shorter parts of the sequence that actually align to each other. So we'll end up with, some optimal local alignments that are very high scoring, right? And get back to a 0. But then we don't have to consider all of this other space that is sort of not important to understanding the relationship between these two sequences. So it's a way for us to identify subalignments. And just as important to note that there are other alignments here, right? Like it's not just we found the one that we thought was the best one, but there's other alignments that are still within this graph that might be important to us. So basically it's the same rules as the global alignment, but no negative numbers when filling out the graph. So as I go through, if 0 is the best number, is higher than any of the other scores, I'm just going to put a 0 in that position, right? And I'm just going to continue to go through and fill out this path, and what I'm going to end up with is a graph that is going to allow me to trace back without any negative numbers, right? And every single one of these zeros then can be a starting place. And so now you would go back through starting from the highest scoring node and find the local alignments that best fit, that are your highest scoring, right? And in this case, we kind of have 2 of them. Our highest node is 4, and so we'll go back across through this and find the best alignments. And notice that one is a little bit longer than the other, so we might prefer one over the other, or we might not. Notice that this local alignment, though, is the same region of the sequence aligning to the same 2 different regions of another sequence. So you can see how this can complicate this alignment process, but that is one of the reasons why we actually do want to figure out a way to evaluate and weight the effects of different sequences. So when we think about sequence alignments, why do we want to do these 2 different types? Well, they have different purposes. So global alignments are really important for when you know the boundaries of your queries and where you think indels actually matter. And this is traditionally what we've used to explain patterns among species, right? Do phylogenetics and things like that. Local alignments, there are cases where we don't know the boundaries, right? And in this case, we're thinking that indels are not important, right? Like we don't want any of those negative values. This is a very important process for finding conserved sequences. So sequences that have been conserved among different species that are likely because of some sort of shared ancestry or homology, or that are functionally convergent or functionally similar to each other. Again, BLAST is going to be using a local alignment approach to do this. So the B and the L stands for basic, local, and the A stands for alignment, right? So that's what we're actually going to be using when we do this type of assessment. So BLAST stands for Basic Local Alignment Search Tool. So we're not doing global alignments, we're actually doing local alignments in this case. Originally developed by a group of scientists at NCBI and Penn State and Arizona. It's basically been used to compare a query sequence to a library or a database of other sequences, and it's one of probably the most commonly used bioinformatics tools out there. What it does is it uses a basic threshold to try to find sequences that it thinks are similar, and it does this by locating short matches between 2 sequences. And this is a really important method for us, right? So it's one of the ways that we can take an unknown sequence and identify it by comparing it to a database of known sequences. This approach, this method is known to be a heuristic method. So a heuristic method is basically an approach to problem solving that employs a really practical solution, but is not always guaranteed to be optimal or perfect, right? It's just going to try to satisfy our immediate needs. And the heuristic approach that it's taking is it's trying to find the highest similarity pair for a short segment of your DNA sequence and a sequence in the database. And once it finds it, it's going to say, well, that's above a certain threshold level. I'm going to hold on to that as a hit, right? And then that hit is then expanded to, as long as it can, to compare the similarities between the two sequences. And then those expanded hits are then reported back to the user, and the best and ranked, right, by these similarity scores, these alignment scores. There's a bunch of different tools within BLAST. There are ways that you can compare nucleotide sequences to nucleotide databases. You can compare protein sequences to protein databases. You can compare nucleotide 6-frame translations, or you can think of this as like mRNAs to protein sequences. You can do proteins to transcribe sequences with tBLASTn, and you can even do nucleotide 6-frame translations, or mRNAs to mRNAs, right? Using tBLASTx. Some other algorithms in there are the position-specific iterative BLAST, which is great for identifying and finding profiles, protein profiles, or family domains within sequences, as well as the delta BLAST, which looks for conserved domains within proteins and uses those to align them. So there's a lot of different components of the BLAST algorithm. We're just going to talk about the general, straightforward nucleotide BLAST algorithm, which is going to function in the algorithm is a fairly straightforward algorithm. Basically, you're going to remove low complexity regions, or regions where there's repetitive information that's not very informative from your query sequence. You're then going to break that sequence up into K-letter words, and we'll come back to this idea a lot in our terms of sequence analysis, but this idea of breaking a sequence up into K-mers, or K-letter words, so K words that are all 5 letters in length, that are 4 letters in length, that are 10 letters in length, that kind of thing. And then you're going to have this list of all possible matching words. You're going to keep only the high scoring ones. You're going to organize these high scoring words into an efficient search tree. And then you're going to repeat steps 3 and 4 for each K-letter word in the query sequence. So you're going to go through one K-letter word at a time, identify the high scoring sequences, and then organize them into an efficient search tree. Then you're going to take that information and you're going to scan the database for exact matches to these high scoring words, and you're going to extend those matches out as far as you can, as long as the alignment score increases, to generate something called a high scoring segment pair, or an HSP. Then you'll list all the high segment scoring pairs in the database whose values are above a certain threshold, and you'll evaluate, the significance, or the e-value, and we'll talk about how that's done later, but how good of a match, how random, what's the evidence that we've got a good match versus a random match for this high scoring pair. Well, then if we find any high scoring pairs that are sort of within the same region, we can connect them to make a longer alignment, and then in the end, we're going to show the gapped local alignments of each query species matched with the database, and then lastly report every match whose expected score is lower than a specific threshold value. Right? So we go through this whole process. This is the algorithm. This is the means by which we're actually generating those matches that you might see if we were to go back and look at this, right? So this result comes from us taking these steps. And so this is the entire algorithm. Within it is this matching section of scores where we're using scores to find these high scoring segments, right? These alignments, these local alignments to generate these high scoring segments. So that's these Swift-Waterman local alignments that we're generating, right, are just part of this broader algorithmic approach. So that's a basic way of how to do a sequence comparison, how we do pairwise sequence analysis in BLAST. And I think it's just good to know that we have this, some of the key things to remember. We've got this path that underlies what's going on through this process, right? That we have these rules that are going to determine the alignments that we're going to have to compare between different alignments, and that there is both a scoring matrix and a set of rules that we're going to be using to generate the final result that we want. So this is a first example of how we would take an algorithm and apply it. And I think, just understanding that there are these rules, understanding what they are, understanding how an algorithm works, and understanding how computers can leverage these simple rule systems to be very effective ways for us to do data analysis is really important.